This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 269, recorded on January 24th, 2014. Hey, everybody, I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today here in the TWiV studio, Dickson Despommier.
Hello, Vincent.
How are you, Dickson?
I'm very well, thank you.
I haven't seen you all week until today.
It's true.
Which I must admit is wonderful.
Oh, you mean missing me or seeing me?
Not seeing you.
I didn't know how to take that.
Where have you been?
I've been jaunting about. We took a trip to India over the Christmas holidays and stayed there until January 4th. And then I took a trip up to Bard College, which is the reason why I couldn't make it last week.
So you haven't been on TWiV since India?
That's correct. I think that is right.
You know, you sent me a picture of a polio thing, a bobber.
I did. Yeah, it was
a
in
sign
Uti.
saying,
It was in Uti.
Uti.
Yep, that was in the hill stations.
Nice.
Hill stations, yep.
Dixon, we have a full crew here today.
I know. This is exciting.
The entire TWiV crew is here.
I tell you.
And joining us from North Central Florida is Rich Condit.
Hi, everybody.
Did you do what you had to do today?
Yeah, I did what I had to do today.
You're interviewing students, right?
Yes, I just finished right before this. I interviewed. I've got the admissions office trained to confine my interviews to 1 o'clock and 2 o'clock, so I can do TWiV at 3.
Excellent.
So I did that. We did Viraholics at 12. Where all the virus labs get together and if somebody gives a presentation, then I interviewed 2 students, and here I am.
What did you call it? Viraholics?
Viraholics.
Yeah.
Hey, that's a great title.
You integrate
There you go.
genomes.
Viraholics.
Wow. Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
You back from the Biff?
Yeah, I don't need to go to the Biff at the moment.
That's nice.
It's a beautiful day, isn't it?
Beautiful day to go to the Biff.
Let's talk about the
Yeah,
weather.
we didn't do weather yet.
Come on,
but
we will.
It is. Let's see here. It is -8 Celsius.
Nice.
Barometer's falling. Outside temperatures. Outside humidity is about 30%.
You got to get some snow tomorrow.
Yeah, supposed to get a little snow coming in.
So
-8 C.
Yeah.
Got it.
God.
Yeah.
-7 here. It's been like this all week.
It's nice, actually.
It's really cold, and
It's cold.
it's going to stay like this for at least another week.
Right.
Yep.
I have minus temperatures through next Friday.
That's it. Oh, and what about the Super Bowl?
The Super Bowl it happens to be here in New Jersey.
Yeah. And
But whoever talks about it is always saying the next Super Bowl is in New York, and it's not in New York, folks.
It's in Jersey.
And it's going to be really cold.
It will be cold.
But,
By the way, it's 51 here in North Central Florida. It's 10.5 degrees Celsius,
Above
and
Celsius,
I'm feeling really insulted about all of this.
I think that where our other co-host is out in Ann Arbor, Michigan, Kathy Spindler, is probably a lot colder, right?
Hi, everybody. Yes, indeed. It's 7 Fahrenheit, -14 Celsius.
You win.
And
You win.
Wow.
Feels like, where's the wind chill? It's a different app, I guess.
Feels
but anyway,
cold.
it's pretty cold. It's colder than where you
right.
guys are.
That's right.
At
Wow.
night, what does it get down to, -18 or something? See?
Probably. I haven't been paying attention because I'm not out at night. But I walked in this morning. It was fine.
You're not a vampire, huh?
No, right.
Here, well, I have this app that tells me the temperature, every couple of hours. So it gets down to -17 at like 4 or 5 a.m.
Goodness.
here. So you have to be colder.
Have you?
You must really bundle up to walk in, Kathy.
Yeah, but last week I overbundled, so today I got it just right.
Oh, good. Do you have snow on the ground where you are?
Yes.
And doesn't
Yes,
it sound funny when the temperature gets down around there?
Yeah.
No, it's not like it is normally, right?
No, it's right. It's very noisy.
Crinkles.
When I drive out of the driveway,
That's exactly right.
it's cool.
Yeah.
All right, here we are. What do we do now? Oh,
I
we
think
do
we
a
get to
TWiV.
start this show.
All right, so Ashley from last week was going to be here again, but she decided that this week's question better goes to TWiP. So, Dickson, just to fill you in,
Yes.
we're teaching this Coursera course, and she gives them a discussion question every week, and she promised them that she would bring some of the best responses to TWiV.
Oh, good.
So we did one last week, but the question she posted has to do with parasites, so we'll do that on TWiP. So we have to do a TWiP next week, okay?
You bet.
All right, we have a follow-up from Samuel who writes, Dear Vincent and TWiV team, thanks a lot for the help. So Samuel asked us to advertise his meeting in Normandy in April. We did that last week. By the way, Stéphane Blanc from the cauliflower mosaic virus papers you discussed is one of our invited speakers. How about that?
Cool.
Small world.
One of the papers we discussed is one of the speakers at this meeting.
How about that?
We'll happily advertise TWiV in Roscoff. All right. Cheers, Samuel. Thank you. And Trudy writes, Hi, TWiVers. I'm writing mostly in follow-up to episode 268 in response to Toph's question about the possibility of a virus that triggers an immune response upon reinfection but without protection from disease. I would like to suggest RSV as such a virus. First, prior infection, especially in infants and young children, does not guarantee a safe response to later infection, often allowing an infant to become symptomatically infected more than once, even within a single RSV season. Second, studies with the formalin inactivated vaccine of the 1960s show that viral challenge after immunization with this vaccine actually triggers a rather severe Th2 type inflammatory response, which manifests itself as enhanced disease. So the immune response is there, but so is the disease upon second exposure to viral proteins. In fact, this is one of the reasons why 50 years later we still don't have an RSV vaccine.
Yeah, I was shouting as I was listening to you guys. I was shouting RSV into my iPhone. Okay, that was my pick. But of course, I wasn't there.
And
Yeah.
So I
this
agree.
just to make it clear, this is respiratory syncytial virus,
Right.
not Rouse sarcoma virus.
Right.
Yeah.
Right.
Big problem.
I would also like to send in my own version of a do-it-yourself virus. So the last 3 weeks we've had knitted viruses on TWiV,
yes.
Dickson.
Yes.
This virus was made by my husband, and he actually used it to propose marriage to me. At the time, I happened to be doing research on RSV at the CDC, and knowing my love of viruses, he made a replica of an RSV virion out of styrofoam, complete with wooden and plastic glycoproteins on the outside.
Wow.
The virion has a removable cap showing the inside, which is lined with glass beads,
How could you say no to that?
representing matrix protein, nucleic acid made out of pipe cleaners, and a felt heart on which he placed my engagement ring.
Oh, my goodness.
It is completely based on a picture he found on the internet since he is not a scientist. I think this is probably one of the geekiest proposals I have ever heard of, but I loved it. Thanks again for the great work. So her photo is lovely,
It's terrific.
and we will
That's
make
great.
it our image of the week,
It
our
is
episode
adorable,
image.
Adorable.
I want to point out that one of the
Yes.
major glycoproteins is a golf tee.
Golf tee.
Yes, it certainly is.
Cool.
Yeah, it's pretty neat, Trudy. How can you say no, right?
That's right.
No, that's terrific.
Unless, of
That's
course, you got
wonderful.
the glycoproteins wrong or something like that, and then you could say, not sure I want to marry this guy. He doesn't really know much about glycoproteins.
I mean, Rich, it would have been funny if he had made the other glycoprotein golf balls, right?
Then Rich would have really been teed off.
It's really terrific.
You can't see the image, Kathy?
No, I can't. I don't know if it's a firewall thing with our Gmail or what. But since you're going to make it the show image, I'll see it then. Just like
Yeah,
everybody
Google's
else.
having problems today.
They are.
It was why we couldn't use Google Hangout to connect, and I don't know what's going on.
Maybe their server went down the biff.
Yeah.
That's right.
I
Maybe-
will email my son right now and see if I can get the word on this.
Good luck on that.
Yeah, do that. And it's probably because of the Nest.
Of course, his
thermostats.
email address is Gmail, so
Right. All right. Now, today we have two papers on a very similar topic, and I've been wanting to do this for a while. And it has to do with sensing of DNA by the innate immune system. And we have talked, obviously, last year, I think, we talked about this wonderful CGAS paper where we showed that we discussed the paper by, what's his name, Rich?
James Chen.
James Chen, showing that cyclic GA synthase, GMP-AMP synthase, is a sensor of DNA in the cytoplasm, makes these cyclic molecules which act as second messengers and turn on interferon synthesis. So the problem here is to sense nucleic acid as foreign and turn on the interferon system.
Right.
Okay, Dickson?
Yes.
Now, at the time, this was 2013, I complained that most DNA viruses don't dump their DNA in the cytoplasm, they go to the nucleus. And how to deal with this. It has to be a nuclear sensor. Now, we immediately received several emails saying there are nuclear sensors or nuclear sensors.
Nuclear.
Please,
Got the nuclear first.
no.
No?
It turns out that a linguist that I listen to thinks that the word is evolving toward nuclear.
Oh, no.
No.
And the reason is because of a parallel with the word molecular.
Oh.
Wow.
Yeah.
It's a fun.
No, I think it's because we're evolving towards stupid.
Toward Biffs.
Yeah.
Wow.
There's all kinds of evidence about evolution of language, and it's really not right to talk about what's right and what's wrong.
I refuse to believe this.
I'm with you, Alan. I hope I'm dead when this happens, because I don't want to be there.
Well, they'll end up spelling it differently eventually.
My family all says nuclear, so I've made my contribution.
Yeah.
Good.
But I agree that, language does evolve. We all understand that. And it evolves in ways we can't control,
That's
but we
not
can
how
still
it's
have
spelled.
we can still have opinions, right?
Not how it's spelled.
Transfection is not all DNA into cells, but that's what everyone thinks. So I complained, and in fact, there have been sensors of DNA in the nucleus discovered. I found a paper in 2011 where they described it. So I feel really stupid because I've been complaining that no one found a nuclear DNA sensor, and they in fact
unchallenged. You were unchallenged until then.
So, but I'm very willing to admit that I screwed up.
So what are the nuclear sensors?
Well, this one that we're going to talk about today is called IFI-16.
Listen, I didn't know 10% of what I've learned since I started doing TWiV.
Yeah, I know.
Okay, there's a lot of stuff out there to know.
Yeah, but I get very opinionated, and I look ridiculous. It's just got to be a nuclear sensor. So,
That's a good way to test opinions, though.
I
throw them out
suppose.
there, and people say, you're full of it. And then they explain why.
So two papers today were sent independently by two people as back when we did the CGAS paper, and I've been wanting to do them, so I thought we would do them together today.
Good.
And the paper that so the protein is called IFI-16, interferon-induced protein number 16, and it was originally identified as a cytoplasmic sensor of DNA, but then in 2011, in a paper in May in Cell Host Microbe, it was identified as a sensor in the nucleus of Kaposi's sarcoma-associated herpesvirus DNA. So it goes way back.
Wow.
See, now nobody calls it a nucleus, do they?
Nucleus.
That's right.
No, they don't. So the two papers today are related to this, and the first one is called nuclear IFI-16 induction of IRF3 signaling during herpesviral infection and degradation of IFI-16 by the viral ICP0 protein. And the authors are Orzali, DeLuca, and Knipe. And David Knipe is a well-known professor at Harvard, and Neil DeLuca is a well-known professor at the University of Pittsburgh, both herpes virologists. And this is published in PNAS. So IFI-16 is this interferon-induced protein number 16, which, as I said, has been shown to be a nuclear... I can't even say it now. You guys screwed me up.
No.
Nuclear.
You're doing this. I'm watching you do this.
Nuclear. Come on, let's get it. Nuclear.
Yeah.
Nuclear.
Nuclear. And I guess I'm going to be president one day, right?
Yes, but of what?
Ooh, that was quick. You should go back to India, and you get quick when you go to India.
You
Dickson.
do. You have to be quick over there.
And in these two papers, you will see nice evidence that it is a nuclear sensor for sensing DNA. Both papers use herpes simplex viruses, which are enveloped viruses with a very large double-stranded DNA genome. They get into the cell and go right to the nuclear membrane. They dock and they put their genome into the nucleus.
So
I think that's astonishing all by itself.
Yes.
Here.
You know,
Here.
you get this transit through the cytoplasm without exposing the genome to the cytoplasm to any significant extent, and deliver this payload to the nucleus. I think that's wonderful.
Yeah, but isn't this title a little bit misleading according to your original definition of sensor molecules? Because doesn't the sensor molecule induce interferon?
Well, it leads to the induction of interferon.
So what is this? The title of this paper implies that this has been induced by interferon.
So nuclear IFI-16 induction of IRF3. So IRF3 is a transcription protein through which IFI-16 and many other sensors work to turn on interferon synthesis.
Turn on. Okay, fine.
So the sensor, it could be IFI-16 or RIG-I, which senses RNA. They sense the nucleic acid. Then there's a signaling pathway which leads to the phosphorylation of IRF3. It dimerizes. It goes in the nucleus. And then it turns on the transcription of an interferon.
Got it.
In other cytokines, okay?
Okay.
So that's what that... And in this paper, they're also looking at the antagonism of IFI-16 by a viral protein, which is like an added bonus here that we get.
Okay.
All right. Is that okay?
Sure.
So I just want to point out that I wasn't alone in my problem with sensing in the cytoplasm. They say in the introduction, an interesting paradox in DNA sensing involves herpes viruses, which are a large class of double-stranded DNA viruses that replicate in the nucleus. It's unclear how they are sensed in the nucleus during infection. Are you already bored, Dickson?
Stop it. I'm allowed to yawn.
All right. So then they talk about IFI-16 being reported to be a cytosolic DNA sensor, but in some cells, it's actually nuclear. So this is very interesting. So the original report, it was in a cell that happened to be cytoplasmic, but in other cells, people found it in the nucleus. So that's why they did this study, in part. So they use herpes simplex virus type 1 in this paper, and they use wild type virus later on, but initially they use 2 viral mutants, and it's important to talk about how they differ. All right, the first mutant, which they have called D109. All right, this is a replication defective virus. It has mutations in what are called immediate early genes. All right, and the gene expression pattern of herpes viruses is a cascade. You have the virus gets in, and then you have immediate early genes expressed, and then some of those are responsible for the next wave of expression, early and then late.
Sounds like adenovirus in some
Yes.
DNA viruses generally do this. They have
do
a
this.
schedule of gene expression.
See, RNA viruses don't schedule anything. They just
They just go in and
Do it all at once.
go in and wipe the cell.
They're more ad hoc.
Yeah, and in fact, that sequence. Have we done this in Virology 101?
No, we haven't. I don't think so.
Okay, because that
yes,
pattern.
I'm sorry, we did. Yes.
And what cell
We
line
did.
are they working with here? This is in vitro?
So this is human foreskin fibroblasts.
Okay.
Dixon is asking good questions here.
Yes.
Well, I haven't
This
been
is
around
good.
for a while.
Why do you want to know what cells they're working with?
Well, because you said some cells have cytoplasmic sensors and some have nuclear sensors.
Oh.
Which ones do you pick?
Well, they picked one where it's
Yeah, but if
nucleus.
everybody worked with cells that only had cytoplasmic sensors, they would have missed this.
Yes.
Yes.
So that's the way science is. It's
So maybe that's
serendipity.
why you have that opinion that nuclear sensors don't exist, because the only results that you were familiar with was the ones in which, et cetera, et cetera.
Yeah.
Sorry.
Human foreskin fibroblasts, which are cells you can get a lot of.
Yep.
And you can get them primary.
Yep.
So the one mutant is lacking, D109 is lacking 5 immediate early genes, or they have mutations in them. So you get no viral gene expression. So this virus gets in, the DNA is there, and that's it. So you can just measure sensing of the DNA.
Got it.
The other virus has mutations in a lot of these, but has one immediate early protein made, ICP0. which if you know anything about, this is like the mantra of herpes virus, simplex virus, ICP0. It's an immediate early protein that gets in and turns on the synthesis of many, many later proteins.
It's a transcription factor.
It's a master regulator.
Master regulator. Then late in infection, it goes out in the cytoplasm and does things, and it gets packaged into virions.
Wow.
It's really... So you have basically two viruses, one without any gene expression, the other with ICP0.
Right.
You okay?
Yep.
Right. The two viruses really just differ in whether or not they have ICP0.
Yeah.
The
Right.
106 is missing the same four other immediate early proteins as 109.
Right. So they infect cells, these human foreskin fibroblasts, and they look at the readout initially is an interferon-stimulated gene, which happens to be ISG54.
And how do they assess the induction of interferon?
They measure the mRNA by real-time PCR.
Okay.
So what they see is that, you compare the two viruses, the one without any early gene expression, you get nice induction of ISG54. But the virus that makes ICP0, you don't see much induction of this interferon-stimulated gene. So it suggests right away that 0 is interfering.
So if you had another viral infection that you were not responding to very well, could you use this mutant herpes virus as a stimulator to boost your levels of interferon?
Yeah, but who's going to give herpes as therapy?
This doesn't result in an infection, though.
I don't know. I don't think it would be
You
approved.
could count that as a vaccine.
It's a tough sell, though.
I think it's a tough sell.
Is it
Actually,
SELL or CELL?
Actually, if I'm not mistaken, this D109 is the basis for a herpes vaccine that David has been working on.
David Bloom.
No, David Knipe.
Yes.
I think that's correct.
It could be.
So it would have to be defective.
So,
to
Dickson,
use it.
your strategy, though, is
Yes.
good. Maybe you could have a small molecule that did the same thing.
Right. Now, have they broken down the genome of herpes? I guess this is in the paper, too. But if you keep chopping it up, how far down the genome can you go and still get induction of interferon?
All you need is the DNA.
Now, but how much DNA?
Well, in the next paper, they put pieces in.
Yeah, exactly.
But you don't need any gene expression. That's the thing.
No, I got it. But I was
Actually, there's
just curious.
one of these sensors that is activated by a 7-dimer.
Yeah, it's a vaccinia DNA piece, right?
Right.
Yeah. Yeah, that's very interesting.
And okay.
All right, so now there's a little experimental issue here in that these two viruses have a different particle to PFU ratio. I think we've talked about that before on TWiV, right?
Right.
And so one of them basically has more defective particles than the other. So they're delivering different amounts of DNA to the cells, so they think that this could influence their results. So they spend some time dealing with that. And basically in the subsequent experiments, they have to adjust the multiplicity of infection to compensate. I don't want to go into it because it's not all that important, but
It all works out.
it all works out in the end.
Yeah,
Right. One of them has more defective particles, so you have to put more of that virus on the cells.
to deliver the same amount of
Deliver
DNA.
the same amount of DNA. Yeah.
Right.
So then with this compensation, they do a series of experiments where they infect with the two viruses, and then they do a time course where they measure expression of interferon and interferon-induced genes. And the pattern is similar again with the virus with none of the DL109, which has none of these 5 immediate early genes. You get very nice response, which is presumably sensing of the viral DNA. And then the virus that makes ICP0, you get much less.
So what turns them off?
What do you mean what turns
How do
them
you
off?
stop producing interferon?
Well, there are proteins called suppressors of cytokine signaling, which is part of that. The
viruses induce that also?
They might. The interferon, the way the transcription induction of interferon is set up, it's actually a rather rapid. It turns over. It's built into the system. It's quite interesting.
Okay.
It is. That's a good question.
Okay.
Yeah. You don't want to have it firing all the
Of
time,
course
right?
not.
Yeah, a lot of things are like that.
And what happens to the DNA of the herpes, the defective herpes? Eventually degraded, or it just sticks around?
I would guess there's no infection, so it would go away eventually. But their experiments are over by then. I mean, they don't go very far. There's 6-hour time points here,
Got it.
right?
Got it.
So then the next experiment is kind of cool. When the herpes the herpes particles dock on the nucleus, apparently there's a cleavage event of one of the viral proteins that is needed for release of DNA into the nucleus. Okay, it's a proteolytic cleavage, so they just treat cells with an inhibitor of this protease.
Wow.
And then they do this similar kind of experiment. They asked these two mutants what happens to ISG54. And in fact, the proteinase inhibitor blocks the induction of interferon-responsive genes.
Neat.
So the interpretation of that is you do indeed have to deliver the DNA to the nucleus in order to get this interferon induction.
Exactly.
Right,
Yeah.
so that's cool.
Yeah, I like this experiment.
I guess they did that with
Right.
DL109 only.
Yeah, the way I think of this is that you can't do these. Basically, the ICP0, the comparison between ICP0 and DL109 or D109, or D106 and D109, the plus and minus ICP0 tells you that the virus, pardon me, Jane, knows what's going to happen. Okay, and has evolved ways to step on this response. So you can't do these experiments with wild type virus. There's too much going on.
Right.
So the multiple mutant is a way to just deliver the DNA to the cells without any immunomodulatory response from the virus. And the ICP0 virus is a way of showing that this is actually relevant, that if you allow expression of this virus gene, it'll shut this response down. It's very cool.
Yep.
May I interrupt this program just for a moment to indicate that there's a bald eagle flying directly out of our window right now.
Wow.
How can you tell if it's a bald
It
eagle?
has a white tail and a white head. And it has wings that are much broader than almost
What
the
is
entire
it doing
body.
here? Did you know there were bald eagles?
I
here?
saw two of them going down the river earlier this morning, and I said, God, those are bald eagles. And the eagles are making a big
Hey,
comeback.
it's huge wingspan.
Very huge.
It's like 3 feet, right?
It's a bald eagle.
Okay. Well,
Sorry.
you have white hair and white tail, too.
This is all true.
So far we've shown that if you just deliver herpes DNA to the nucleus, you get an interferon response. So now is IFI-16 involved? Well, of course, in today's day and age, we knock down IFI-16 with siRNA, right? Mm-hmm. And indeed, that attenuates the induction of interferon-stimulated genes. So IFI-16 is necessary to activate the induction of interferon-stimulated genes. Now, some of you may remember Sting.
Well,
Well, from the 80s.
I met him once.
I was waiting. I was giving you a little lead there.
Had a good time with him.
Sting is a singer.
Yeah.
And
He's also a world wrestler. He's a world WWR or whatever that thing is, wrestling.
actually, in your Vertical Farm book, Sting had a quote on the cover, Dickson.
Yeah, that's true. He agreed
But
to
this
say
Sting is a
something.
cellular...
And Frodo's
I got it.
sword, wasn't it?
That's right, yes, which glows when orcs are near.
Yes.
Very good.
Yes. This
We
week,
digress.
popular culture.
Do you have any idea of how he got his name?
Who, Sting?
Yeah.
It's not his real name?
No, he showed up one day in a black and yellow T-shirt that was striped horizontally.
Yeah.
And I think the drummer
bee
looked
Sting.
at him and says, "Guy looks like a bee."
But speaking of black and yellow,
That's how he got it. Yeah, black and yellow. Go
that's
on.
me.
Yes. Vincent Black and Yellow.
Sting is a cytoplasmic protein that
Okay.
is a sort of a bridge between IRF3 and the protein kinase that phosphorylates it, which is in response to sensing some nucleic acid in the cytoplasm, either by C-gas or any other of these sensors. There's a whole bunch of sensors in the cytoplasm.
So I have another question then. I'm sorry, we were
It's okay.
finished with
It's fine.
the nuclear pores that you're talking about here. That's just one kind, right? And it needs a nuclear peptide signal in order to import proteins into the nucleus.
Right.
That's the same pore that we're talking
That's right.
about now. So
That's
that's
right.
the same receptor that you're talking
Yeah,
about.
so
The protein
the
portion
herpes
of the-
particle
Got
docks
it.
onto that pore.
I got it. And
then the DNA goes in the nucleus.
No,
It's very cool because the particle's too big to fit through the
but
pore,
of course,
right?
But of course.
So Sting is an intermediate, and they say, well, it could be that's an intermediate for IFI-16 signaling as well, right? So they say, is Sting important for this interferon response? And they knock it down with siRNA. And the answer is yes. Now, is anyone worried about this? Sting is a cytoplasmic protein. Like, do you have any concerns? remember, they're thinking the sensing is going on in the nucleus, right?
Exactly.
But sting is in the cytoplasm.
So you were talking earlier today about leakage from the nucleus back into the cytoplasm of a small portion of DNA that might be extruded into the nucleus. Is that a possibility?
That's a good question. In the next experiments, we're going to look at where IFI-16 actually is in the cell.
Okay.
I got to say, and they comment on this, that the knockdown of sting is not really robust.
It isn't.
It's less than twofold, and yet it has a dramatic effect on the interferon induction.
Yeah.
So that's curious, I suppose. Interesting.
Right.
Yeah, I mean, so the key here is that the idea is there's nuclear sensing going on, yet this cytoplasmic sting is still important for turning on interferon-stimulated genes. So there's no answer to that in this paper, but we'll get to that in a minute.
It also means that if your knockdowns look like they're not working all that well, go ahead and do the experiment anyway.
Mm-hmm.
Right. Yeah.
That's a very good thing.
And jumping ahead, they have a nice model slide, and this connection between IFI-16 and sting is a question mark.
Yeah, that's one of the unanswered questions. Yeah, how the sensing of DNA by IFI in the nucleus, how does it engage sting in the cytoplasm?
Yeah, because we'll get there.
So the next set of experiments, they look at the localization of IFI-16 in cells, and they show that it is indeed in these human foreskin fibroblasts. It's in the nucleus. Right. Immunofluorescence.
And stays there during infection with all these viruses.
That's right. It doesn't change. I guess there's some previous reports that there are some cases in some cells where the localization changes, and we'll see in the next paper how that works. Very nice connection.
So that's why we've got IFI-16 in the nucleus, we've got sting in the cytoplasm, and somehow they're talking to each other, but we don't know how.
Right. And the second half of these experiments is to look at sting. And in fact, sting is in the cytoplasm, as you would expect, and it doesn't change either.
Yeah, because IFI-16 said, "Don't stand so close to me."
Exactly. Exactly true.
We have a show title.
We do.
Don't stand so close to me.
We do.
Wow. I'm just amazed. All right, so the idea here is that you have sting in the cytoplasm, IFI-16 is in the nucleus. How are they talking? Well, maybe something's exported.
This might be a protein-protein interaction.
Yeah, something is exported through the nuclear pore, right?
Right.
So they have a drug which inhibits nuclear export by a certain pathway. It's called the CRIM1 pathway for CRM1. All right, and so they treat cells with leptomycin B, and indeed, this inhibits the what is their readout? Phosphorylation by the kinase that is phosphorylating IRF3, sorry.
Wow.
Okay, so signaling requires nuclear export. So something's coming out. You're right. It could be a protein. It could be something else.
Correct.
In this case, we could call it a factor because we don't know what it is.
Here.
But you better not if it's a protein.
No.
I like looking at you, Dickson, and preaching. We're preaching.
I'm following this.
You got it so far,
Sure,
Dickson?
Absolutely.
All right. Now we move into ICP0. And they are saying, I think there's some implication that ICP0 previously has been inhibiting IRF3 phosphorylation. So they do some infections of their cells with the two viruses, D106 and D109, and then they look at they compare the amount of phosphorylated IRF3. They can do that readily. You have antibodies that are specific for the phosphate. Okay, and what they find is that in cells that express ICP0, which is the 106, right?
Yes.
Mm-hmm.
They have less IRF3. Actually, not just phosphorylation, but accumulation. There's delayed accumulation of IRF3.
Right.
So at 6 hours, in the DL109 infected cells, which don't make ICP0, they see phosphorylated IRF3, but not in the cells infected with D106 reduced levels.
They've separated nuclear and cytoplasmic fractions, so
Right.
you can see the IRF3 moving from the cytoplasm to the nucleus in a phosphorylated form.
Right. So the idea is that actually 0 is somehow sequestering IRF3.
So between the virus and the cell, there's been a nuclear arms race.
Has been.
Very cool.
And so they do a series of experiments to address that. And they show using this antibody that, in fact, having ICP0 there, you're early in infection, helps sequester IRF3 there.
Figure 7 is lovely.
It's a beautiful
Yeah.
figure. Yes.
It's just that's probably framed on somebody's wall somewhere.
Right, so you can actually see, using indirect immunofluorescence, you can actually see
Right.
that ICP0 and IRF3 at least transiently co-localize in the nucleus.
Cool.
So it's like the ICP0 has gone and sought out the IRF3 and sopped it all up in these little balls.
Yeah.
Right.
And by the way, all of the papers that we're talking about today are open access.
Nice.
Right.
So people
Yeah,
can
so
go
you
look
can
at
look
these
at these
figures if they want.
Yeah, the pictures, there's lots of immunofluorescence in these papers, and they're really
Yeah.
pretty.
Yeah. In fact, the next set of experiments is similar. They use both Western blot analysis and immunofluorescence to look at the levels of this is now IFI-16, the effect of ICP0 on IFI-16. Before we were looking at IRF3, so a little indirect, but now we're looking at IFI-16, and in fact, the presence of ICP0, you have a reduction in the levels of IFI-16. That's by Western blot, and then if you look by immunofluorescence, you initially see that in fact IFI-16 co-localizes with ICP0, which suggests that the two proteins are interacting, having some kind of intimate interaction. And then it goes away. The IFI-16 signal goes away. So they think that somehow ICP0 is making IFI-16 degrade.
And that's not too far-fetched because ICP0 is a ubiquitin ligase.
Right.
Right.
So,
So-
ICP0 has this activity which would cause a protein to be ligated to ubiquitin, and then it would be targeted for degradation to the proteasome, which is a massive degrader in the cell.
Indeed.
Right.
I have another-
So it grabs it and stuffs it in the trash.
Another basic
Right.
question then is that a nuclear factory is created by herpesvirus.
It
right?
is. It is. Yeah.
So it all occurs in the nucleus. How does interferon work then?
Well, that's way downstream of this paper, but there are many proteins induced by interferon, which
Yeah,
have
and this signal
Oh,
gets out of the nucleus.
Okay, so this is an early
sensing.
sensing of an innate immune response, which could result in an importation of a protein from the cytoplasm into the nucleus that would interfere with
Could.
the herpes factory.
Sure, why not? Absolutely.
How many? Well, I guess there are multiple.
But remember, the virus is antagonizing this response, so
exactly.
It doesn't even get
like
to that
it.
point.
It's fighting back with
It
an
doesn't
anti-
even. Yeah, it doesn't even get to that
Oh, I
point.
got it. But in the mutant that doesn't have this anti-phosphorylation protein, then what would be the effector molecules from the cytoplasm that might interfere with their replication in the nucleus?
Well, that's a good
In
question.
your opinion.
I don't know offhand because there are over 1,000 of these interferon-induced molecules.
That's incredible.
So we just answered that question for polio in the past year,
Uh-huh.
and we out of 350 of these molecules, 18 turned out to inhibit polio.
Oh, yes, I remember that.
Right, and
I do
then
remember.
this has been done for a number of other viruses, but I'm not sure if it's been done for herpes yet. So, all right. So they show that ICP0 has this domain called a ring finger.
Not a zinc.
So my colleague here, Sol Silverstein, worked on herpes
Right.
viruses for years. And he had people working on ICP0, and I just remember hearing about this ring finger every year, which is basically a cis-his-cis motif, which coordinates two zinc cations and makes a finger, basically. And that just is the way it interacts with other proteins. So they can alter this by mutation, and they can show that, in fact, if you get rid of key residues here, so it's no longer ubiquitin ligase, you don't lose IFI-16 anymore. So it's nice evidence that it's ICP0 is promoting the degradation of IFI16 in a proteasome and ring finger dependent manner.
I want to know what the phenotype of that virus is.
Which one? 10?
The ring finger mutant.
Yeah.
Okay? Like, is it... gee, I don't know how I would assay it, but I would think that it's not... it's defective in countering this pathway, right? So it might be...
Yeah.
Would it be interferon super sensitive or something? No,
Yeah,
not necessarily. I don't know, but something.
Might not grow well, but it might also impact other things, right?
Yeah.
Besides
And that's in a
interferon.
wild type virus instead of this guy that's knocked
Yeah.
out for all those other immediate early genes, so
Right.
it's going to be complicated.
Yeah. I mean, yeah, exactly. I suspect the ring finger is involved in other things too, but
Yeah.
I mean,
Their summary figure is terrific.
yeah, it's a really nice, clear summary figure. Shows how the virus gets into the cell by endocytosis. It docks on the nuclear pore. I don't know if you can see that, Dickson.
There's a little glare, but I can see most of
Okay,
the
and then the
detail.
DNA goes in the nucleus. It
Yep.
gets bound up by IFI16, which then leads to the production of something that goes out in the cytoplasm that helps
Right.
sting
It's a question mark that goes out to the
Yeah,
side.
a question mark
Yeah,
goes in the
Question mark,
cytoplasm.
that's right. That's right.
Does something with sting, which allows IRF3 to be phosphorylated, which then come back in the nucleus and turn on interferon. But in the meantime, ICP0 is made, which is antagonizing IFI16.
Amazing.
So in the end, herpesvirus wins. So the question is the big question. What is that thing that's going out? And they say in this paper, we're working hard on it.
Yeah,
Exactly.
for sure.
Exactly.
I was
Yeah.
thinking, I wonder if you could assay for that thing using a setup that's similar to what James Chen used to isolate C gas.
I'll bet. Sure.
You know, where you have some cytoplasmic fraction where you can use to look at phosphorylation of IRF3 and some fraction from infected cells that you can add to that and see that happen and then purify it out of the infected cells, something
Look at
like
that.
that.
Hardcore biochemistry.
Yeah, hardcore biochemistry.
All right, go ahead. Sorry.
I also noticed that a lot of people seem to have adopted your flying turkey for the ribosome.
Is that one in here? Oh, yeah, look at
Yeah,
that.
look at that.
Nice.
I saw it somewhere else too.
It is absolutely a flying turkey. Look at that.
It is.
Look at that. Yeah.
And I wanted to point out, in case any of my students are listening, that the herpesvirus DNA goes in and they picture it first as its linear form, and then, of course, it has to circularize.
Yeah, it's right there accurate because, of course, this is a herpes lab, so they would be accurate. Yeah. Now, there are two things I wanted to point out here. First, in their discussion, they talk about how it's been unclear how herpes virus DNA is sensed because how herpes virus DNA gains access to the cytosolic compartment during infection has been unclear. And they say some have proposed that the DNA may be made available to the cytosolic compartment by the degradation of the capsid. So this is, I had trouble with this, but our colleague Steve Goff just came back from a innate virus meeting at Keystone, and he said a lot of people are showing that, in fact, you get leakage of viral DNA. Leakage on the
Right.
way in.
Right.
Yeah, that wouldn't surprise me at all.
So-
None of this is perfect.
Yeah, that's what you always say. None of this is perfect. It's biology is sloppy, right?
Yeah.
The other question is, how does the cell distinguish herpes DNA from its own DNA?
Roger
Yes.
That's
Yeah.
that's a good question, right?
Yeah, that's a good question.
And they talk about how, so when the herpes viral DNA comes in the nucleus, it's chromatinized, right, it's wrapped up in histones, and they say, but the way it's wrapped up is different from host cell DNA, so maybe that's part
And
of
it
it.
may take a little while. It happens pretty fast, but at least transiently, it's not chromatinized, so that distinguishes it from those.
Maybe
cells.
that too. And they also say that DNA may be nicked or gapped, and that could be providing a binding site for
So
IFI6.
is there
Well,
a-
and it's initially linear, so you have exposed ends.
Yeah.
And what is known for sure is that herpes virus infection stimulates a DNA damage response in the cells.
Right.
And then the DNA gets all glommed up with a bunch of basically nuclear stress proteins that put it in these little compartments and trying to get rid of it. And part of that is the DNA damage response, and then the virus fights back. So there's a transient period in the beginning of the infection where it's clear that the cells are recognizing that there's some foreign DNA that's been put in there, and or something's wrong DNA-wise, and it's trying to deal with it. But then the virus gets past that.
So does this virus also attack cells at a certain stage of their division cycle? Or does it matter? It doesn't matter at all as to what stage of the cell cycle it's at before it goes in.
I don't know.
Really? Come on, Rich. I thought you knew everything.
I don't know. No.
Because as cells undergo mitosis, obviously, and that's why we were going to get to the next idea, I guess, right? Because when the DNA becomes spread out and distributed without a nuclear membrane during cell division, if things bad happen to our own DNA as well as to the herpes, would be damaged.
So you're saying if herpes virus infects a cell, puts its DNA in the nucleus, if that cell then undergoes mitosis, there's a period when the nuclear membrane is gone, right?
That's right, because these guys only did 6 hours worth of experiments, right?
Yeah.
But what if they did 24 hours or 36 hours?
Well, the fact is that on the single cells where the DNA comes in, they respond. So
are these all synchronized?
I don't.
Well, it's a primary. It's a primary culture. So
right.
Are they even dividing?
I don't know.
Yeah, it could. Quite often,
they're just
when I do these kinds of experiments, I'm doing them on an established monolayer where probably most of the cells are dividing.
Just thinking about this is.
And I'm still thinking about what distinguishes the viral DNA from the cellular DNA, and just being in these PML bodies or ND10s may be enough. Just the location may be enough to help it get distinguished, and by the time they're there, the IFI-16 is gone and degraded?
Could be.
It's not integrated, right?
No.
No, so it's not latent or anything like that.
Well, it is latent, but it can be latent, but it doesn't integrate.
Right.
Okay. Shall we move on?
I'm sure we'll hear more about that.
Oh, yeah. Absolutely. This is cool stuff.
Yeah, as they say, currently represent a high priority for future studies. That's the
Yes.
yeah.
Yep.
Significance number 2 on the grant.
Second paper is also in PNAS, was published around the same time, June of 2012. The paper we just did is also a 2012 paper.
Right, October.
And this is called Acetylation Modulates Cellular Distribution and DNA Sensing Ability of Interferon-Inducible Protein IFI-16. And the authors are Lee, Diner, Chen, and Christea. This is from Princeton University. And I think this was sent to me by one of Lynn Enquist's virology students after I said they need to find a nuclear sensor.
Yes, I remember that.
And they said,
there was a letter, right.
Here it is. Okay, so in this paper, they also use herpes and a little bit of vaccinia, which will make Rich very happy. But not virus, just a piece of DNA, I think.
Well, anything will do,
And they mentioned that they talk about IFI-16 and how there's evidence that it's predominantly nuclear, but it's also been reported in the cytoplasm. And so they want to look at this and whether it can sense infection. So the first thing that they do is simply take a look at the sequence of the protein, IFI-16, and look for a nuclear localization sequence, a relatively short sequence of amino acids that allows a protein to interact with the nuclear import machinery and get into the nucleus. And indeed, they find a couple of them. Motif 1 and motif 2 and motif 3 and 4. They find four different NLSs of different sequences. And so they simply remove them from the protein. They put a GFP label onto IFI-16, and they remove each of these NLSs and ask what happens when you put a DNA encoding this into a cell by DNA-mediated transformation.
Perfect.
Guys, and what happens? So they find that these motifs 1 and 2 are both needed. If you take either one out, it doesn't localize to the nucleus, and the other 2 don't seem to be important.
And that's pretty striking.
Yeah.
Here again, they got great pictures. and it's really pretty striking, the effect.
All right. So then they ask, is the localization of this protein important for sensing herpesvirus DNA? All right. So they infect these cells, they infect cells producing, no, not expressing, they're producing either the wild type IFI-16 or their NLS mutants. All right. And does someone remember what virus? Are they using wild-type virus? Yeah, I believe
so, yes.
Yeah. And so they look for the viral DNA, and if it's near IFI-16 by fish, they go fishing, Dickson.
I like that.
Fluorescent in situ hybridization.
Yeah, I know it's fish.
It is?
Yeah.
Well, I have to say it for our listeners.
It's true. This is true.
And that's why you could see in a immunofluorescent view where the DNA is, and then you
If
can
did
also
it in Drosophila, it would be fly fishing, wouldn't it?
Ooh, that's nice.
Not bad.
You're listening.
Of course I'm
Yeah,
listening.
and then they can look for where IFI-16 is by immunofluorescence as well. So basically, when they infect, they can, well, first they transfect, I believe, right? No, they infect first, and they see that wild-type DNA gets in the nucleus and co-localizes with wild-type IFI-16. But if you use either of these motif deletion and IFI-16s, they don't co-localize with viral DNA because they don't get in the nucleus, of course.
Once again, beautiful pictures.
Really nice experiment.
I'm just amazed that you can light up the DNA with a fluorescent hybridization probe, and light up the protein with an antibody and see co-localization in the nucleus in these cells. It's just amazing. Yep. And again, the results with the motif deletions are the nuclear localization motif deletions are striking.
You'd thank a jellyfish for that.
So they also put viral DNA in by transfection, and they use, I think here they're using pieces of herpes DNA, if I remember. Yeah, DNA fragments. And again, asking if they're co-localizing with IFIs. And the interesting thing here is that there is some co-localization in the cytoplasm, because when you put DNA into cells by transfection, that's where it goes first, and then it goes into the nucleus. So there is a reduction in nuclear co-localization, but there's initially some in. Even these motif deletions that don't get in the nucleus, you can see cytoplasmic co-localization.
The other method that they do here is to do an infection and then, if I got it right, immunoprecipitate the IFI-16 and look to see if there's DNA bound to it.
That's right.
Okay, it's like a chromatin kind of like a chromatin immunoprecipitation experiment.
And those are the ones reported in the graph, because you can get numbers for that. Percent DNA binding, yeah. So you can see the motif deletion. When you infect, the motif deletions knock out DNA binding completely.
So I really like that this kind of approach where you have two completely different approaches that converge on the same answer.
Right.
That's very convincing.
Right.
And there's another approach in this figure where they are looking at a promoter, interferon beta promoter, driving a reporter as a readout of induction of interferon. Mm-hmm. And they ask, what's the effect of deleting the IFI-16 motif? And you can see that in a wild-type IFI-16 scenario, the infection turns on the interferon beta promoter. And if you delete the motif, it does not. So it's suggesting, again, that nuclear presence of IFI-16 is important for sensing viral DNA. You okay, Dickson?
I'm very okay with this.
All right, so now they do a really cool set of experiments where they purify IFI-16 from cells. What kind of cells are they using here? CEMs. So CEMs.
T lymphocytes.
T lymphocyte.
Okay.
T lymphoblast-like cells, because they abundantly express IFI-16. Okay, and it apparently is also in the cytoplasm in these cells, because if they put this 70-mer DNA of vaccinia virus into the cytoplasm, they get a response that's dependent on IFI-16.
So my sense is that this stuff didn't kind of come out of the blue.
Oh,
Okay,
yeah,
they've got that different cells express different fractions in the cytoplasm of the nucleus, and they also have some background information that suggests that potential post-translational modifications of a nuclear localization signal could affect that, so they go looking for post-translational modifications of their nuclear localization signals.
So herpes disappears into the nervous system, yes?
Ultimately, yeah.
Yeah. So
simplex, yeah.
What do those cells look like with regards to
That's
these?
a very good question. Good question.
Whether they have this at all is
Don't know. That's a
debatable,
really good question.
because in general the interferon response in the CNS is muted, because it's not a good thing for cells that don't divide.
Right.
Right. Interferon is not really good for you. It can hurt you.
I hear that.
Right. Now, here's what they do. They isolate endogenous IFI-16. I'm going to read this because this is so cool. They digest it in gel or in solution with trypsin or GLUC, analyze it by nanoliquid chromatography coupled with mass spec with two complementary fragmentation techniques, collision-induced dissociation and electron transfer dissociation. Got that?
I got it.
Got it.
I'm not sure I understand it.
That's my
Basically, they want to know if any of the amino acids are modified in any way. And they basically identify 6 phosphorylation and 9 acetylation sites.
Wow.
Most of
Boy, I
which...
got a protein for these guys. I'd like to see. I'd like to see this done with a protein we work with. Get all the phosphorylation.
Yeah, this is
sites. Nice job.
This is a mass spec lab.
Yeah,
So
a great
Cristea-
job. This is a big protein too, and it's got a lot of modifications.
Right. So it turns...
Mass spec is really, really spectacular in what it can do these days.
Yeah, it's fabulous. It's in high demand. So they basically show that the... All right, do you remember the 2 major nuclear localization motifs that they previously identified earlier in this paper?
1 and 2.
1 and 2?
Exactly.
Yeah.
Each has an acetylation at a lysine. There's a lysine at 99 and 128. So that suggests that maybe this is regulating, right, nuclear import. And that's what they show. They basically change these lysines to something else, and they show that that changes the nuclear abundance of the protein. So apparently acetylation is interfering with nuclear localization, because if you change the amino acid so it can't be acetylated, this thing gets into the nucleus very well.
Right.
They show that by one of the assays is an in vitro assay where they take nuclei out of cells and put them in vitro, and you can look at import. And also they look at it by fluorescence, EGFP approaches. So
Yeah, they substitute the wild-type amino acid with other amino acids that will mimic acetylation. Okay, so they can have things that kind of sort of look acetylated or not, and check and look at their localization.
So what is acetylating IFI-16? That's the next question. So they actually use a inhibitor of a histone deacetylase. Now it gets a little bit tough with the negatives here.
Yes.
Yeah.
I did a paper last week with histone deacetylase inhibitors. I got so balled up. For 10 minutes I was trying to figure out the results. I had to edit it all out because it was just... And 3 of us, we couldn't figure it out. Anyway, so trichostatin A is an inhibitor of histone deacetylase. So a deacetylase takes off acetyl groups, right? And that lets the proteins go in the nucleus. So if you
inhibit a deacetylase,
right? What's
you
that?
allow acetylation to proceed.
That's right. And so you're going to block nuclear import.
Right.
And that's in fact what they find. This histone deacetylase inhibitor blocks nuclear. It compromises nuclear import. So the suggestion is that histone deacetylases, which exist in the cell to, modify histones and control transcription, are involved in the localization of this protein. It's pretty cool, right?
Very
Yeah.
good.
I think it's neat.
Yep.
I think they have some evidence for what is acetylating IFI-16. This is new to me. So there's a P300 is a known transcriptional coactivator. Is that correct?
Yeah, and I was wondering if we're really talking about the same thing here or not.
It is. Apparently, there's something associated with P300 and then the other protein that's bound to it, CBP, that has histone, has acetyltransferase activity.
All right.
And this is the P300 involved with the RB protein, right, Kathy? And all that, yeah. So there's something there that is... So that's not surprising that a transcriptional activator would have a acetyltransferase, right, because want to modify the histones, right? So apparently that it may be doing something with IFI-16.
Right. To make it clear, I'm pretty sure it's P300 itself has the hat activity. They talked about
it does, yeah.
The purified P300 hat domain has the ability to acetylate K99 in vitro. So, yeah, it itself is
I found no other reference to the acetyltransferase activity of P300. I thought it was associated, but it is. It's known to have acetyltransferase. Yeah. Okay. All right. So that's basically the result. So now you can get IFI-16 where you want it by acetyltransferase. So how is that regulated? That's the cool question, right? And one thing I'd like to know is, does it change in response to virus infection? Does the cell change it depending on where the virus is? Does the virus antagonize it? Lots of interesting questions here.
And once again, they have a terrific summary figure for this whole thing. Right.
One thing that confused me just a little bit is in the abstract and the very last lines of the discussion, they talk about their two-signal model. And I never really quite get what is signal 1 and what is signal 2. They don't spell it out, and I need it literally spelled out.
Well, it's with or without an acetyl group.
You think that's all it is?
Yeah, that's it. That's all it is.
All right.
Yeah.
Okay.
That's it.
I like in their summary figure, they illustrate, they signify signaling with a little icon like you get on your wireless signal.
Yeah.
Yes, IFI-16 uses Wi-Fi to detect double-stranded DNA.
Nice.
That's how it works.
That's pretty good.
Is that 4 bars or 3 bars?
It's got 4 bars.
I got 4 bars. That's
4
heavy-duty
bars.
signaling. Yeah.
That's pretty good.
Oh, actually, it's only 3.
It's 3.
1,
And
2,
maybe that's what needs to replace the question mark in the other papers as well.
There you go.
Can you relate any of this biology to the fact that some herpes infections can result in carcinomas?
I couldn't offhand, but why not?
I mean, it plays with the acetylation.
I don't know if it
program.
plays with it. No, all we know is that acetylation is important for the localization of IFI-16.
Right.
Whether herpes plays with it, we don't know.
And we don't understand the mechanism by which they actually induce cancers.
Herpes viruses?
Yeah.
I would go back to the time we had Pat Moore on the show.
Okay.
Yeah.
I think about that show all the time because he pointed out that there's a relationship really between the immunomodulatory functions of viruses and causing cells to proliferate abnormally, because the antiviral proteins also are cell cycle control proteins. They're all twisted up together, so when the virus goes and tinkers with those, a consequence of that is to tinker with the cell cycle.
One correction. Apparently, it's been known that acetylation has effects on nuclear localization. It can either promote or prevent nuclear import, depending on the context. Okay. So in this case, acetylation is preventing, but apparently there are other examples where it promotes. Nothing is black and white.
Nothing.
Okay.
Except the color's black and the color.
Yeah. And black and yellow, right?
That's
Yes.
black and yellow.
Let's read some email. Sorry, Dickson, you don't have your iPad.
That's okay. I'm a good listener.
You're a good listener.
I'm a very good listener.
Alan, can you take the first? No, I think Rich should take this first one. Sorry.
Yes.
Rich.
Okay. Let me get down here. Da-da-da-da. Mark writes, "Hi, TWiV team. Happy New Year 2014. I'll be brief. This image has been spreading through the internet today. It is time sensitive, given the cold weather front that has been punishing the eastern United States. I received this special customized Florida image from a cousin who lives in north central Florida, and I immediately thought of TWiV, your collective interest in the weather, and I wondered if Rich Condit shared my cousin's description. It's good fun that appeals to both genders. Of course, you can't read from the image during a show without being branded with an explicit tag in iTunes. However, I'm sure you can evolve like a virus and convey its essence without triggering an immune system response from those who rate podcasts. Is that sufficient challenge to stimulate your collective creativity to read this on TWiV? All the best, Mark. And he sent a cartoon
that uses the word fudge an awful lot.
Yes.
Right. Fudge.
The U.S., Dickson.
It divides up the U.S. into various segments and talks about how cold it is.
Yes.
Right? Using the word fudge a lot, including the modified one down in Florida that uses a different modification of this. Says, "Oh, fudge."
I got it.
Okay.
And then California is out to the left, and all it says on it is LOL.
Yeah. Right.
This is great.
Yeah.
I see where you are, Kathy. You're in the same. No, you're in a different place from us. Yeah, New Jersey.
She's cold as fudge.
Right. Yeah. I
Or-
think this is so cool.
Fudge. It's close.
You know,
So, Rich, does this characterize North Central Florida?
you know, people around here are really touchy about cold weather. So they always
why they're
flip out.
there.
But frankly, I don't think this year's any worse than any other.
Right.
You know, we always dip down to somewhere close to freezing. We've brought all the plants inside. We got plants covered. There's a lot of harangue about the cold. But, you know, there's always a few days when it dips down below freezing, and it's not all that big a deal. Yeah, it's cold today, but I rode my bike in. It's not a big deal.
Well, the citrus farmers are worried, right?
Actually, they always worry. It's a big deal, and in fact, it has not been as bad as forecast. Last night it was forecast to be like freezing for a couple of hours, and they missed it by 5 degrees. It was like
That's why my
it didn't get down to
That's why
freezing.
my grapefruit tastes like ethylene glycol.
There you go.
So, Rich, whenever I ride in and it's pretty cold, people will say, isn't it too cold to ride? And I say, it's a lot like skiing, and you're not out for as long. But
That's good. That's good.
I have to tell you that my neighbor across the street has been riding. I saw her come home last night with her bike helmet, light, and everything. So she's got you beat.
All right. Oh yeah. I'm not. I have my limits.
I was in Princeton yesterday, and I walked past the football field, and there were guys out there in shorts. They were shoveling the snow so that they could do something. Like maybe they were lacrosse players or something. The guys were in shorts. It was minus.
I thought their SAT scores were higher than that.
Well, no, if you're exercising and doing something really active,
that's
like
true.
if you've ever gone running in the cold, for the first few minutes it's really, really cold, and then it's comfortable.
Yeah, but it's a good idea not to expose skin, though, right?
No, that's true, but there was a football
Yeah.
game. One of the playoff games was up in Green Bay, right?
Yeah.
And it was San Francisco against Green Bay, and the temperature was 6 degrees above zero.
Yeah.
And they were breaking out in sweat.
You know, they get paid a lot. They can be
They were
cold.
no. They were worried about overheating.
Really?
Yeah, absolutely.
Kathy, next one's an adenovirus email.
This is from Lance. He writes, Dear TWiV crew, I love the show, love what you all are doing for science. I graduated with a PhD in microbiology and immunology and worked on host virus interactions of HCV. I'm currently working as a virologist in a public health lab, and I wanted to pass along an interesting finding. A couple weeks ago, we confirmed an adenovirus infection of the genitals in a male patient. My mind was blown. I was always taught that adenovirus was a respiratory virus. A link to a manuscript of a large public health study is here, and he links to a paper from 1995 where Heyerholzer is the senior author. I think it's important for people to remember, especially PhDs and MDs, as we branch out and do other things outside the lab, that some microbes have lesser known presentations. Thanks again. So I took a look at this paper, and Mike and I conversed about it. They show adenovirus in these various sexually transmitted type of disease locations, but they don't show any causality. And it's maybe not surprising. Adenovirus has been linked to cystitis, so finding it in some of these locations isn't all that surprising. And then one of the ones that they find a little bit more prevalent is adenovirus 37, and that's the one that causes the eye infections. And then Mike pointed out how the virus could get from the eye to those nether regions, and I just said that was too much information.
Yeah.
But
Yeah.
Come on, Kathy, we're all biologists.
Yeah.
here.
Yeah.
so he said it was a consequence of him sitting in on too many infectious disease lectures. So
Yeah. It's like.
Well, I'm glad to hear you say that, because adenovirus really gets around. These different serotypes cause a lot of different, infect a lot of different tissues and stuff. So I wasn't too blown away by this, so I'm interested to hear your spin.
Kathy, any of these serotypes, GI ones?
I have to pull the paper up again.
They say 37 is the eye. How about 8?
7. No, the GI ones are 40 and 41.
Okay. Because it's very easy to get contamination from the GI tract down in the genital area. That happens with E. coli all the time, and that's what causes these extra intestinal E. coli infections. But
Right.
these are not the right serotypes.
Right.
Yeah, I didn't know about causality here, but this is an old paper. I guess it hasn't subsequently been shown.
Yeah. So, and it's only, these were found in 0.33% of their patients. I mean, they had 7,000 patients, so they had a real number, but 23 out of 7,000.
Yep.
Yeah.
Nevertheless, the correspondent's message
Yeah.
is valid. Some microbes have lesser-known presentations. Keep it in mind.
Right.
Right.
Alan, can you take Michael's?
Sure. Michael writes, "Hello, Vincent and TWiV gang. Good morning. It's 8 a.m. in my part of London, and it's 3 degrees Celsius, and humidity is 92%. First, I'd like to say thank you so much for the effort you and the gang put into making the Twilogy. I have listened to and enjoyed all the episodes you've produced. In the following paragraph, please excuse my language. My love of evolutionary ecology is not matched by my ability to put it into words."
Is Dickson listening?
I'm listening.
He doesn't mean
I'm
language
listening.
that way.
Listening.
He means he's I've just listened to TWiV 261, and there was a statement that bothered me. One of your contributors said that there was a universal rule in biology that parasites do not go back to being free-living. This statement bothers me because if it is true, then it should not just apply to parasites. Any organism that evolves to be specialized for a particular habitat will lose genes that allow it to use other habitats, and gain genes that allow it to more effectively exploit the now narrower niche. So I guess I'm saying I don't know if specialization is irreversible, but if it is, then it must apply to all organisms, not just parasites. The question is, therefore, do we have any examples of facultative parasites which largely depend on the host for growth and reproduction, or are there obligate parasites that we have not yet discovered culture conditions for which are capable of reproducing? Culture conditions in which they're capable of reproducing. I would be grateful to hear your thoughts, and especially those of Dickson. Merry Christmas. Oh, thank you. And thanks for all the hard work. We're
Appreciate
behind on the
that.
emails.
Well, in the parasite world, in the worm world at least, there is one transitional organism that we've discussed on TWiP in the early days called Strongyloides stercoralis. And it has a free-living cycle. That is to say, it can produce both male and female worms, and then they mate in the soil and produce offspring, which then go on to become either male or female worms, and they can reproduce. And so as long as their food supply is in abundance, the moment their food supply starts to run out, which in this case happens to be the deposit of feces, and as soon as that starts to dry out, it causes a shift in their biology where all of the worms then stop development at the third stage, which is the infective stage for humans. And for other mammals as well. So that's the stage that then penetrates the unbroken skin and becomes parasitic. Here you've got clearly an organism that can go both ways. Whether that tells you much about how long it's been around or what kind of a parasite it really is underneath all of that machinations is still up for grabs in terms of, I guess, an evolutionist. A strict evolutionist would be the person to discuss this in more detail with. As far as losing genes when the pressure to keep them is removed, I would beg the same question for resistant microbes that become resistant to a certain antibiotic, and then if you stop using that antibiotic, how come those organisms don't revert back to the wild types that they were before the antibiotic was used? And the answer, because it's expensive to keep a new gene around, right?
Well, often they have fitness costs, so they don't last either.
Right.
So this statement was made by Jean-Michel Claverie in the Giant Virus TWiV. He said, it was in the context of, his idea is that viruses started out in cells and then they moved out and then they lost genes, right? And he said and he was saying, the first thing that probably goes is DNA replication genes and then transcription genes and then, translation genes go first and then the other. So his point was, once you lose those, it's very difficult to get them back.
Yeah, right.
And so he sort of made a generalization to all parasites, all obligate parasites. Once you lose a gene and you become an obligate parasite, you don't go back again.
Interestingly enough, though, a lot of these worms have actually acquired genes on their way to becoming parasites from the free-living form, which is the Caenorhabditis elegans archetype.
So Dickson, are there facultative parasites besides the strongyloides, which you said is an intermediate?
We have a term for them. We have an obligate parasite, right? And we have commensals, which live in the same host but don't cause any harm.
Can they live separately? No, they can't.
Of course
right?
not.
No.
Of course not.
So there's no example of someone who can live in a host, who needs to live in a host, but then could live on its own again if it had to.
Right.
Well, I think there's some fungi that do that, but not eukaryotic parasites.
Viruses, obviously.
At least not that we've found yet.
I haven't found any. That's
right.
always
That's
the caveat, yeah.
I'm sure we're going to get 20 letters on this. Oh, I worked on this on my...
Well, and I would contest this notion that that genes are all that expensive to keep around.
Well, that's one of the arguments that they use. But.
Right, but I think if you look at genome sequences that we have, there's a lot of cruft in there. There's commonly stuff left over that's not being used currently that was probably useful at one time.
Yep.
Or we don't see it being actively used right now. And evolution as a process tends to be kind of additive. So if you really need a particular trait, you'll adapt the material at hand to develop that trait, or you'll be wiped out. And if you don't need that trait, well, depends on how much it takes to keep it going. If it's just a question of keeping the gene around, then that's not a big deal. But if it's something really some structure like an eye that's really energetically expensive and prone to infection, then you're probably going to lose it if you don't need it if you live in a cave.
So there are bacteria that have now symbiotic relationships, and they've lost genes, so they can't live on their own anymore. Right inside those pill bugs, there are bacteria that help the bug get nourishment, and they in turn get stuff from the bug. So they can't go back. They've lost the genes, eons ago, and that's it. Right.
They can't easily go back. There'd have to be some kind of a step that they would go through to regain those functions.
They'd have to regain all those genes. Yeah,
that's right.
Yeah, but I think we're probably looking at extreme cases here.
That's true. I mean, the Rickettsia are sort of a transitional zone between they're looked at least they used to be, between the obligate intracellular parasites, and these are all intranuclear, by the way, and bacteria species, which you can then culture apart from the host. So can you culture Rickettsia? And the answer is, under very, very unusual circumstances, you can, but it requires a very rich cell-free environment for these organisms to reproduce. The same can be said for malaria parasites. You can culture them outside the red cells, but they're extremely difficult to get to reproduce, and you only get 1 or 2 cycles before they run out of steam. So it's not that we can't do it, it's just that we haven't invested a lot of our energy and time in replicating the conditions under which a lot of these organisms need in order to succeed. And I agree with Alan. I think that the argument that they're expensive depends on the environmental situation, right? They have backup systems for emergency purposes, for changes in weather or changes in temperature or humidity or pH or all kinds of things, that there are genes there that will help them do those the transitions, like the heat shock proteins. They're not active all the time. They're only active at certain times under stress. So a lot of this must relate to the specific situation rather than the general situation.
Right.
Also remember that this is in the case of viruses, the gene loss is one of of several hypotheses.
Yeah, right.
Who knows which is right? And that happened to be Clavery's idea, but
These could be rogue genes from normal organisms.
I mean, some people feel that viruses started small and acquired genes. Okay, in
that's
fact, one of Eugene Koonin is one of the proponents of that, and hopefully we'll have him on a TWiV in the future, and we can talk about that. But I think from what we can see, who knows? I would say, it doesn't look like they can go back, but there are always exceptions.
I guess
And also, for something to exploit, to back out of a niche that it's in, there has to be a niche that it can exploit better.
Yes, that's correct.
Sure. So I have
That's
another
correct, yeah.
general question that you could ask anybody at this way. You take all of the DNA virus sequences, right? Take them all.
Just DNA virus?
Yeah, just DNA. That's all.
Okay.
And ask how similar are any of those DNA viruses to any normal DNA that we can find in any living organism?
Some of the proteins, like DNA replication machinery, is similar.
But you assume they robbed that from the cell.
Well, that's the big question.
That's the question. Yeah.
Right,
Right,
because
but-
it could be they robbed it from the cell, or it could just be that if you want to replicate DNA, you've got to converge on a particular solution.
Sure. So the question I guess I have is what is the percentage similarity between the DNA virus that best matches with any other organism? What is the percentage match on that situation?
I don't know offhand. So if you take a DNA polymerase of a virus, how similar is it to a host DNA polymerase? I think there are segments that are similar that are parts of the active site
Sure.
or some
Of
nucleotide
course,
binding.
Of course.
I think for the most part it's dissimilar, but there is enough to say these clearly had a common origin.
Got it.
That's
Okay.
all I know.
All right.
Yes, Kathy.
Just going to say, this week in vocabulary, I didn't know the word cruft.
Yeah,
And
me
it
too. I had to look it up.
comes from 1958, students frequenting the MIT Tech Model Railroad Club, and just means garbage. It's kind of
Junk.
a computer software term.
Yes.
Oh, that's
Might
the
be
word.
related to crud or crust or so forth, but
Cruff and Biff.
There are a couple of
Cruft.
different stories. Yeah, a
Cruft.
couple of different stories of its origin.
Don't
That's-
forget fudge. The fudge is in there. Got to be in there someplace.
Cruft and Bidge.
It's got to be a code word for the
Cruft,
fudge.
Fudge, and Biff. That's today's episode. One more is from Todd, who writes, if you haven't seen it yet, the Twitter feed Real Scientists, one word, is a rotating Twitter account holding fantastic discussions with followers. It's a curated account that rotates between submitters every week or two. The end of December was a scientist trekking through a jungle. This week it's a fantastic discussion of science and communication. Cool. Check it out, Dickson.
I will.
Unrelated question. Can you pronounce the full name for C. elegans?
Cenorhabditis.
Yes, of course.
yes, and Dixon just did it a few minutes
Did,
ago.
yes.
Yeah,
I was
I noticed
was
that.
thinking you must have read ahead, but it.
I
didn't
once
have
asked.
any to type.
I used to teach here, of course, as everybody knows, in parasitic diseases. But I would often ask the student body, how many people here know what C of C. elegans stands for? And about half the hands would go up, and the other half says, "I just call it C. elegans."
No,
interesting.
we've all
Interesting.
been saying cenorhabditis for many years.
Yeah, that's right.
See
I'd be
no
like-
rhabditis, hear no rhabditis.
That's right. Cenorhabditis.
And yes, Kathy.
I was just going to say E. coli could be the same thing. How many people
Yes.
know what the E stands for?
Exactly.
Can you say it, Dickson?
And then to spell it,
Can you say E?
it's Escherichia.
Escherichia?
Escherichia or
So I've heard
Escherichia.
Escherichia or Escherichia.
I heard
Which is
Escherichia.
right?
I grew up with Escherichia.
I grew up with Escherichia.
Here you
Really?
go.
Yeah.
With
Really?
Escherichia as well.
So I think this
But
is
I-
the title of a Gershwin song, don't you think?
You say Escherichia, yes.
But Escherichia might be more Germanic.
Oh, yeah?
All right.
So either is acceptable?
I think so.
What other words can we think of that are tough to pronounce?
Oh.
Yeah, nuclear.
Right.
Okay, on that
it's
note.
Abdomen
Yes, let's-
or abdomen?
Abdomen.
There's a whole bunch of
Well, those
things
are
like that. You
those
say
are
potato, I
things.
say
Talk about science. Dixon, do you have a pick of the week?
I do.
What is it?
It's a video. It's a very curious video. It's based on an invention, and this invention is how to grow food in very small places. It's part of the vertical farm scene, and the single unit is called a Volksgarten. Volks garden, Volks garden, all one word. And you can go on YouTube and you can see a high school biology professor a teacher who bought one of these units from the company Omega Grow Systems, and he says how fantastic it is. It's got a light down the middle. It's a big circular device that you can grow stuff on the inside with, and it rotates the whole barrel of this grow system through a nutrient solution. It's very cleverly designed, all right? So the company that makes it has made an animation of what could happen if you apply this single Volksgarten to an enormous building, which they would call a farm dominium, based on the concept of a condominium. And to see this thing work is like looking at a scene out of the movie Metropolis back in the 1939.
I was thinking The Matrix.
Oh, and The Matrix too. The Matrix is pretty good for that as well. But it's not as well illustrated as The Matrix. But it looks like The Sorcerer's Apprentice gone good, because finally they're producing something from an automated a way of doing the same repetitive motion again and again and again. And it's really quite an elegant solution to a very difficult problem. That is, how do you grow a lot of stuff in a very small place?
It's cool.
Yeah,
I like the video. It's neat.
I like it.
Yeah.
Okay.
Okay.
All right.
Thank you, Dickson.
You're welcome.
Kathy, what do you have for us?
I picked an article from MBio from the January-February edition. It's by Arturo Casadoval and Joe Handelsman. And do I have the title of it here? I'll just go to the link I put in. I have the whole paper sitting right here. Presence of female conveners correlates with a higher proportion of female speakers at scientific symposia. It's one of those papers where the title says it all. They analyzed 460 symposia involving 1,845 speakers in 2 large meetings sponsored by the American Society for Microbiology. And not surprisingly, having at least one woman member of the convening team correlates with a significantly higher proportion of invited female speakers and reduce the likelihood of an all-male symposium roster. So it's kind of like a duh, we've known this for a while. But,
right.
It's nicely documented. And in fact,
We've known this in quotes. I mean, there weren't data, but you could look at examples and now here are your data.
Right. There was a visiting eminent virologist once, and she was talking about a meeting that she was going to be convening, and she was starting to list some of the speakers that she was thinking about inviting. And I said to her, "Are there some women you can invite?" And she said, "Oh, that's a really good idea."
Isn't that interesting?
So the fact is that we all have these biases, but here again, if you have some women involved, they're more likely to think about inviting women. So it's important for, up-and-coming scientists to see, I think. So It's a nice, short little paper. It's open access. It's because it's MBio.
That's true. That's true.
Cool.
I once actually co-chaired at a Gordon conference, and we were very conscious of that issue and made sure to issue invitations for attendance and for giving talks in an attempt to keep the numbers fairly balanced. But it was difficult because, as you know, Kathy, there were fewer people in science that were female at that point than there are now even. And I think the numbers are still not adjusted according to the actual population out there.
Right.
So.
Well, at least in terms of graduate school enrollment, there's in most places more women than men. But as you advance beyond that, then
Yeah, if you throw out medical school as a graduate school, because there are a lot of women enrolled in medical schools right now.
Mm-hmm.
We have about 50-50 here. 50 men, 50% men to 50% women.
Alan Dove, what do you have?
I have a website of a scientist turned, I think, full-time photographer now. It's sandgrains.com.
Oh, wow.
This fellow, Gary Greenberg, is a microscopist, and he looks at sand grains, which you wouldn't think would be all that interesting, but they're absolutely fascinating. And these are mostly pretty low magnification things. He does some other stuff too, like flowers and such, but the sand grains are just I found absolutely fascinating.
They're beautiful. Yeah.
And he's doing 50x, 100x. It's fairly straightforward stuff, but he chooses his sands and arranges them, and then does these photos. And of course, the stuff that's in sand is not all uniform. You've got these little, remains of marine organisms and different shapes and different types of rocks that went into it.
Colors.
I mean, there's so many different kind of sands out there too, right?
Yes.
The sands of time, Dickson.
That's true.
Yes.
The sands
This
of
is really
Sands to the
cool.
hourglass.
sands of
He
Las
has a book.
Vegas.
He has a photo book.
Yeah.
Right?
This is very interesting. He worked on the first Superman film where he transformed human pancreatic cells into the planet Krypton.
Yes.
I don't understand that.
I have to
think-
look at the film again.
Islet cells or assigner cells?
Doesn't say. That would make a difference. You're
It-
right.
Oh, tell you about it. You know, it's a whole solar system.
Very
the other
nice.
This really is amazing. It's cool.
one. By the way, as an aside here, just a totally different aside.
Okay, you can do it. It's fine. Just
Please?
this once,
Please,
okay?
okay, this is I was watching one of the Blue Ocean episodes with David Attenborough, and he said, "I'll bet you I can't do a very good David Attenborough, but he says, I'll bet you that you were never aware of where the sand of the sandy beaches comes from. Let me show you." And he showed an enormous school of parrotfish cruising along, munching on coral, all right? And they actually eat the surface of the coral to get the two layers of the coral organisms, and they crunch up in their gut tracts, in their crops, almost like a bird crop, the coral itself, which is then excreted as a fine sand, and that sand washes up on the beach, and that's where all the sand comes from.
Yep,
for these
I have.
atolls. And
I
that's-
have watched these fish do this scuba diving.
Yes, of course.
Yeah.
And so does that's got to show up on these pictures that he's taking of some of those beaches that they obviously got sand from. So it's not really
Probably.
sand. It's sort of like microchips of coral reef.
I'm glad I'm not a parrotfish.
Hey, I'm glad I'm not a coral. Yeah, the coral. I think the curlfish, the parrotfish, sort of. Yeah, they take care of that.
They
before.
deal with it.
They deal with
Yeah.
it. They don't swallow it whole.
They're built for that.
Yeah.
Exactly.
Rich, what do you have?
Well, this is inspired by the fact that Kathy, for the past few episodes, has somebody's made a pick, and then she goes out and buys it.
Not tempted.
Oh.
Not tempted.
So I
You know,
looked
for
for,
this one?
something
Oh.
that Kathy could buy, and I came up with the 2013 Lamborghini Aventador Roadster.
Right.
Okay, and I really got into this,
Right.
okay, because I wasn't, I hadn't really looked at these things before. Kathy, it's a mere $448,295 base price.
Right.
I don't like the way it looks.
Oh,
Oh, you don't?
No.
Oh, that's too bad.
it comes in
Gee,
any color you want.
I was really, okay. Well, I'll see if I can find another one.
Very interesting.
So this, it's a V12, 6.5 liter,
Right.
700 horsepower,
That's crazy, isn't it?
7-speed auto dash CL manual. I had to look that up, and I have
Jeez.
some comments on that. And I'm wondering to myself, who on earth buys and drives one of these things when this morning I open up the newspaper and see that Justin Bieber was just arrested for
Speeding
drag
on one.
racing in his Lamborghini, some guy in a Ferrari in Miami Beach last night. Okay.
Right. Right.
Mildly intoxicated. So there's the answer.
There you
Justin
go.
Bieber buys one
That's
of
right.
these.
So who
Yeah.
owns Lamborghini?
Justin Bieber.
No,
Owns
no.
Lamborghini.
Who
I don't
owns
know.
I think it's Volkswagen.
Really?
I
I
don't know.
believe it is.
At any rate, the one other thing I have about this is this transmission, auto dash CL manual, and
Right.
I don't know the whole term for it. I looked it up at one point, but there's no such thing as a real stick shift anymore.
No.
Well,
don't-
there are, but you have to
Yeah.
hunt.
Yeah.
These are probably paddles where you flip the
right.
paddle. There's
That's
no
right.
clutch.
Just, you're
Yeah,
going
right.
to buy a $500,000 car. I want a clutch.
One day.
Yeah, I mean, so the thing is, for racers, the paddles are clearly faster, but the fun of driving is a clutch and a stick
Yeah.
shift for sure. Yes, I agree that. So my BMW is a 6-speed manual, and I'm sad if it ever goes that it's going to be hard to get another one.
This is true.
So Subaru makes a lot of clutch cars still.
Yeah, you can get most cars, most regular production cars that are not luxury cars are available in a stick shift, but you usually have to special order it these days.
Yeah. I think they're
Okay,
going to.
Kathy, I'll keep shopping. I'll find something for you.
So Morley Safer on 60 Minutes once was asked to drive one of these things back when a new model came out maybe 10 or 15 years ago. And he said one of the big features he found on this car that was not on any other car that he had ever driven was that there were no rear view mirrors. Because you don't need them. He says you
who's
just
going to be behind you?
Exactly.
Gaining.
Ask Justin. He'll tell you.
I have, in addition, one little late-breaking sub-pick here because you guys were complaining about Google crashing earlier on. So I did write my son, who works for Google, and he wrote back, brief outage for Gmail, Google Plus, and Hangouts. Most services have recovered by now, though ours is sadly still coughing up errors here and there. And then, interestingly, he sent a link where you can get, at any time you want, the official status of all the apps at Google.
Right.
It's very cool.
I actually...
That's cool.
Yeah, I checked that one. I did.
So there's
check
still
that.
an outage.
But mine didn't show an outage.
Service disruption, yeah.
I think it says the current status is that service is okay. Over on the left,
Oh, that's
current status.
I see.
But over on the right, it shows that there was disruption.
Yeah, Rich, you should have never told anybody that your son works for Google now because you know what's going to happen.
What's going to happen?
They're going to all write you and ask you, could you please ask your son whether or not my computer is,
Yeah. ... All right. Today, 30 years ago, the first Macintosh was
That's right. It's a birthday.
announced or something. Today's the
It's a
30th birthday of the Mac, and there's a very nice
Yeah.
celebration over at apple.com/30-years.
Right.
Some very cool pictures and videos. And there is a teardown of the 128K Mac over at iFixit. So iFixit, whenever something new comes out, they tear it down. They buy one and they rip it apart so you can see what's in it. So they got a hold of a 128K Macintosh, the beige one in that case, and they took it apart. And this really brings back memories because I had one of these.
I still have one in a closet somewhere.
Yeah.
Really? Save it. Save it. Take it to the Antiques Roadshow.
And it's just so cool. I took some of these apart years ago, and
Wow.
well, anyway, it's pretty. And I have since used Macs, and I love them. And so, check it out.
It's great.
The teardown is
It's nice.
a wonderful, wonderful. I mean, it's all these through-hole parts that you can't even get anymore. It's cool.
We have a couple of listener picks. Peter writes, "Dear TWiV team, I have a couple of listener picks from the BBC. In Our Time, the Microscope. Melvin Bragg and his guests discussed the development of the microscope, an instrument which has revolutionized our knowledge of the world and the organisms that inhabit it.
Good one.
And these are with people from the London Science Museum in Cambridge, University of Leeds. It's a BBC program. Have you seen that, Dickson?
I haven't, but I've seen the first microscope. It's in Holland.
Mm-hmm.
Leuwenhoek's microscope.
I've seen that too.
Yep. It's in Leiden.
Another program from BBC Radio 4, The Next Global Killer, aimed at a general audience, has interviews with people who are working on understanding and detecting emerging zoonotic diseases that could become the next pandemic. Maybe the journalist Alok Shah, who presented that program, could be a future guest on TWiV. I've heard of him. You know him, Alan?
I've heard the name, yeah.
Journalist, author, and broadcaster focusing on stories about science.
Yeah.
Oh, cool. Yes, I've heard of him. Peter, P.S. When will Virology 2 be running on Coursera? It's running. Go over. It's in week 3.
Uh-huh.
Now, for some reason, in the past 2 weeks, 5 or 6 people have sent me, as pics, the glass virus sculptures of Luke Jerram. Richard sent it, and a couple of other people. Yes, Kahau. Hello, TWivsters. Not sure if you've seen this. I thought of you and that it could be a great pick of the week. Love the podcast. I look forward to all the Twixes every week. So he sent a link. And then there were a couple of others. So we'll just stick it in as a pick, although it's been picked before.
Sure.
And
It's okay. You never get tired of those.
You never get tired. And as we say, it's not a problem to repeat because nobody listens anyway. Right? That was Andre Gide.
Or they don't remember anyway.
Right.
Yes, you're just too kind. You ever get mad, Kathy? I mean, really?
Oh, yeah.
Mad? You get
Oh, yeah.
white hot mad?
Oh, yeah.
Yeah. Oh, wow.
Yeah. Don't cross me.
Okay. You can find TWiV at iTunes, twiv.tv. And, of course, if you have questions and comments, you send them by email to twiv@twiv.tv. Don't ask me what TV means, Dickson.
I'm not
I've
going
explained
to.
it 10 times on TWiP.
It's some little island off in the Pacific someplace.
Tuvalu,
Tuvalu, isn't it?
Tuvalu.
That's right.
Right. And
That's
WS is Western Samoa.
Indeed.
Okay. And if you like TWiV, please go to iTunes, which is the other place you can find it, and just leave a comment or a star rating, and that helps to keep us visible. Okay, Dickson?
Yes,
Can
sir.
you do that?
I can. I've done it many times
Thank
in
you.
the past. I shall continue to do it.
Sir. Continue to do it. Kathy Spindler is at the University of Michigan. Thank you, Kathy.
Thanks. This was fun.
And enjoy the cold. Did you put your Schnovel-Wobble thinger together?
Yeah, it's together, and I've used it. And we're supposed to get a bunch of more snow tonight, so I'm going to get to use it again.
So is it really worth getting or what?
Yeah, my brother watched me do it, and he said, yeah, you're not bending your back at all. And
That's a good thing. That's a good thing.
It's pretty fast. My neighbor across the street wants to try it. So
Rent it.
Yeah.
Yeah, I have a Wednesday I have a snowblower I use to clear the driveway, but the blade is a rubber blade and it's broken and it doesn't throw the snow very far, so it throws it like 2 feet. So you have to keep pushing the snow in front. Boy, it was really tough.
Yeah.
Yeah, the blade on mine broke, and now I've found out that the parts aren't going to be coming ever because they don't have them.
Good old John Deere.
Rich Condit is at the University of Florida in Gainesville. Thank you, Rich.
Where it is not snowing, and I don't have to worry about any of this stuff.
You put your time in. Yeah.
That's right. You got alligators in your swimming pools.
Always a good time. Thanks. This is great.
Alan Dove is at turbidplaque.com and also on Twitter. Thank you, Alan.
Always a pleasure.
And Dickson Despommier. Hey, the old Tracaniello.org is gone, right?
No, it's back up again.
Well, now it's back up?
Back up again.
Tracaniello.org.
Yep.
What, so you gave it to someone, right?
Well, I tried to, and then they took a look at it and said, gee.
They don't want it.
Well, not that they don't want it. It's they want it, but they wanted it in a more user-friendly form than they were going to be given it. So it's back up again.
It's a bloody website with pictures of parasites. What more could they ask for?
Yeah, well,
Anyway, Tracaniello.org is
up.
back up.
Still up.
And verticalfarm.com.
Yep.
And medicalecology.com or .org.
It's an org.
All right, Media Maven. Did you write anything for HuffPo lately?
No, I haven't done that. I don't have much to say right now. Actually, there is a new farm opening in Scranton that's 300,000 square feet of indoor farm using the Omega grow systems that I told you about before.
Have you been?
No, but the governor of Pennsylvania went there to help announce it.
Is that Chris Christie?
That's not the governor
Pennsylvania.
of Pennsylvania.
No, I'm just bugging him.
Remember, pencils come from Pennsylvania.
I know. Well, I know you go fishing there.
I do.
All right. Thank you, Dickson.
You're welcome.
Did I leave anyone out? I thank everyone.
Left yourself.
I think so.
Okay.
Except yourself.
And myself.
Thank yourself.
But I
Yourself.
just get distracted here. I just got to make sure I don't want to leave anyone out. I'm Vincent Racaniello. You can find me at virology.ws, Western Samoa.
Indeed.
But you don't have to go there to find
No.
it. You just type virology.ws in your browser, and you'll see what's going on in the virus world. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.